Hi everyone, it's time for us to get started again. All right, so what we're focusing on today is the idea of being multicellular. And as you all know, we are made up of millions and billions and trillions of cells, and so we are considered multi-celled creatures. From an evolutionary perspective, that was a pretty big jump. So we'll talk about the first critters that happened with. Then we'll focus on mutations. Okay, so we'll start to talk about all those DNA molecules that give us our charming characteristics. Then we'll talk about Mendel's first law. Okay, law of segregation. We talked about it before. You have a 50% chance your chromosomes will line up one way and 50% chance the other way. And then we'll talk about Mendel's second law, the law of independent assortment. Okay, and all sorts of other good stuff. So let's go ahead and get started. All right, so let's go ahead and apply some of the material that we have talked about previously. I want you to go ahead and pause the lecture. I want you to read through this, and then I want you to answer it, okay? So pause the lecture, read through the question, and answer it. All right, so as we're going through this should, go ahead and going through the answer, this should make sense. Bacteria do not have multicelled critters. We know that. We talked about them last time. Archaea don't either. Okay, so it eliminates A, eliminates B, eliminates E, and D. All right, so who's left? The eukaryotes. Okay, from a practical perspective, the eukaryotes are the only ones that have multicelled critters. Now, the thing to remember, though, is that eukaryotes can be single-celled. There are several of those, okay? However, they're the only ones that actually are multicellular and complex. So what's interesting about multicellularity and when it actually evolved is it evolved a couple of times, okay? So if we think about, the process through time, billions of years ago is to the right, 4.6 billion years ago, as we know, is when Earth formed. Then you can see the unicellular eukaryotes, and then eventually you see the multicellular eukaryotes, okay? Now, and again, they evolved a couple different times, and if you look at the tree to the left, you'll see the boxes when multicellularity evolved. And I guess it's relatively easy to think about, why there'd be an advantage of being multicelled. Increase in body size, your cells can specialize. What I want you to think about in the long-term trajectory is, why would it be better to be multicelled than single-celled? Because there's definitely pluses and minuses to both ways of life. So you might be asking yourself, what does it mean to be multicellular? And I would say, aha, excellent question. Okay, so there are 3 different traits you have to have in order to truly be multicellular. Write this down. Did I say write this down? Write this down. Okay, the first trait is that your cells have to stick together. Okay, if your cells don't stick together, you're not multicellular. Sounds kind of simple, but it's really true. Secondly, your cells have to communicate. Now, your cells communicate by chemical cues all the time, okay? It's just we don't know it and we're not conscious of it. But that's how your cells actually, get around and do what they're supposed to do and all sorts of other things, okay? So the first one is that they have to stick together. The second one is that they have to communicate. And last but not least, they have to specialize, okay? So your heart cells are only found in your heart, and that's their job. Your skin cells are only found in your skin, and that's their job, okay? So those 3 things are what it takes to actually be multicellular. Once again, write this down. First one is that they stick together. The second one is that they talk back and forth, they communicate. And then the third is that they specialize. All right, so once again, we have another application question, okay? So look at the tree. All right, we see animals are close related to coinoflagellates, and we'll talk about what those are in a few minutes. Just realize that they're single-cell little critters that can also live together in colonies, okay? Then we've got our fungi, which we know also live as multicellular, and we have our amoebozoans. Given this information, what group of species do you think could provide the most information about how this evolved in animals? Now, the trick with this question is to realize that multicellularity evolved multiple times. So it evolved in animals and an ancestor, as well as fungi and an ancestor, okay? Given this information, reading through this question, take a moment and choose your answer, and then start the lecture again. Think carefully. All right, so the answer to this, by the way, is coinoflagellates, okay? It's not fungi because the fungi became multicellular on their own. And it's not the amoebozoans because the coinoflagellates are single-celled critters, okay? So we're going to talk about the coinoflagellates. They're actually fascinating little guys, okay, in the next couple of slides. And the reason they're so cool is this tells us how much multicellularity in animals evolved. All right, so choanoflagellates are these cool little critters that we share a recent common ancestor with, the most recent common ancestor, which, by the way, if you ever see the letters MCRA, that means most common recent ancestor. Okay, so you'll probably see that in the literature somewhere along those lines. So, choanoflagellates, they look like the tiny little cell on the bottom right. They have a cell body, they got the microvilli, and they have a flagella. And you're looking at some real choanoflagellates above that. Okay. What's so interesting about these guys is that choanoflagellates can live by themselves, okay, or they can actually live in colonies. And so they have the option to live one way or another. And it's so cool as far as why they start living in colonies. And we'll get to that in the next couple of slides. So this is one particular species of choanoflagellates. Okay, they live in seawater. They can be swimming as a single cell. They can also be attached to a stalked cell, okay, where they can be attached to a stalk, or they can be a simple multicellular structure that's formed by cell division. And what researchers wanted to know is why. Why do these guys live these three different ways, and what evolutionary advantage could this possibly give them? Now, eventually she would go one step further, and she wanted to know exactly what was it about the prey that would cue in the choanoflagellates to live in rosettes, okay? And so there's a couple of different ways to do this. You can have the live bacteria that you're testing. You can then heat kill them, okay? You can also then destroy a protein that you think is cueing them in. You can then have some destroyed DNA and RNA, as well as some extracted lipids, all sorts of other good stuff. So basically what she did was she took the bacteria and put them in pieces, okay, and asked the same question: what was causing them to form a rosette? If she took those bacteria and then, basically tore them apart and got them into pieces of like lipids and so forth. And the next slide, by the way, lists this all out. So in a second, you'll see this all in one spot. Okay, so again, I mentioned this before. So they wanted to see what was it about the bacteria that would cue these guys in to live multicellular, okay? So they have filtered seawater. And as you would expect, they were not multicellular because there's no bacteria in there. Okay, they then added the live bacteria. Well, they knew that that should cause them to live in rosettes in a multicellular colony because that was what they found before. Then they were curious if they heat killed the bacteria. So this means that they took the bacteria, boiled them, killed everybody, but then still threw them in with the choanoflagellates. Well, obviously they were multicellular colonies, so they still detected the bacteria. Then they took some proteins from the bacteria, destroyed them, probably with heat, okay, and once again, they had multicellular colonies. Then they started to have the DNA and RNA, okay, and then they had the extracted lipids, all right, and so they also had this particular protein, RIF1. All of these, okay, by the way, seem to produce multicellular colonies, which means that the choanoflagellates can pick up on a variety of cues to know that the bacteria are present. And that's actually pretty cool. So they can pick up on the cue of a destroyed protein. They can pick up a cue of DNA or RNA. The lipids, remember, those are fats. All right. Now here's a question for you. Given this experiment, what is the negative control? Okay. So remember, in a negative control, you're expecting your experiment not to have a positive result because you're checking for contamination. So which one of these is a negative control? That would be the filtered seawater. Okay, hopefully that makes sense. If it doesn't, let me know. All right, so then we ask ourselves, what's the positive control? Okay, now in the positive control, you're expecting a positive result. Of these, which ones do we know? Excuse the squeaky toy noise. My puppy is in the mood to play. Once again, downside of no office. Anyway, which one of these is the positive control? Okay, take a moment, think through this, and pick one. If you chose live bacteria, you would be correct. Because remember, what got them even thinking about this was in the presence of live bacteria, they would turn into the rosette. So that better have turned into the rosettes. Otherwise, there's an issue. Now, the RIF1, that's just a protein / lipid, okay? And that seems to be One of the cues in addition to the others. Basically, the choanoflagellates are really good at detecting the presence of bacteria. All right, and when they do, they go into rosette form, and that should make sense because by doing so, they can trap more bacteria, consume more food, and then life is good for them. All right, so as we move right along, it's time for us to think about mutations, okay? So remember, mutations are one of those things that give genetic variation. We've talked about this before through the process meiosis. Isn't it great how all this stuff ties together, okay? And so we'll cover that in the next slide. All right, what I'd like you to do now, okay, is take a blank piece of paper and write down everything you know about mutations. Stop the lecture, put it on pause, go ahead and take a moment and write the stuff down, and then we'll go through the things that I feel are important about mutations. Oh, look, a green smiley face. I got bored with green stars. Okay, so for this lecture, when you see a green smiley face, write this down, okay? So for mutations, you can either add, delete, or change nucleotide base pairs in DNA sequences, and that will create a mutation. This can affect one base pair, this can affect several base pairs, or huge regions of the chromosomes. This can alter coding regions, which actually produce proteins, or non-coding regions as well. And so in coding regions, sometimes you change the amino acid and sometimes you don't. Remember we said before the triplet codons are redundant. You can have a couple mutations and still get that same amino acid in there. Additionally, mutations can be neutral, deleterious, meaning they're harmful, or beneficial. Okay. Neutral, by the way, is the most common source of mutations, followed by deleterious, and then beneficial. So beneficial are the rarest mutations. Additionally, mutations can then be passed on to the next generation or germline, okay? But maybe they're not. Those are called somatic mutations. So write this down. This should make sense. And then as we keep going, hopefully this will make more sense as we revisit these points. Okay, so let's start talking about the different types of mutations. So we can have what are known as point mutations, meaning that they only, meaning that they only impact one base pair. Okay, however, so you're starting with a wild type, which is normal, okay, and then you go through and you can see when you compare the left diagram to the right diagram what the mutant looks like. All right, so you should have done some reading for today. Hint. Okay, hopefully you've done some reading in your textbook, and given that information, you should be able to answer what type of mutation is shown on the right. Okay, take a moment, read through, and then we'll talk about this together. Okay, so let's talk about more depth about mutations, okay? So a missense mutation means that you're changing the amino acid. That's otherwise known as a non-synonymous substitution, all right? Now, a nonsense mutation, hint, that's what this is. That was when you insert a stop codon. And the reason it's called nonsense is basically it stops the production of your protein and it doesn't make sense, okay? Then you can have what's called a frame shift mutation where you alter the reading frame. Now this would be something like an insertion or a deletion where you take that triplet codon and you shift it down or shift it back, all right? And altering the entire reading frame, by the way, is going to have a huge impact on the critter. You can also potentially have an insertion or deletion. Now these are not mutually exclusive because I said you can add or delete bases and that's also a frame shift mutation, okay? Or the last type of mutation you can have is what's known as a synonymous mutation. This means that you have a substitution, but it doesn't change the amino acid. Because remember, there's redundancy built into the triplet codon, where if you have a mutation somewhere, you might still get that same amino acid. Okay, so if you'll recall before, I told you that you need to know how to read this table. I am going to ask you a question on the exam where you have to read this table. But remember, it's open note, open book, so all you have to do is flip to the slide, be able to read the table, okay? So what I want you to be able to do is to realize which codes will code for which amino acids, all right? So realize again, 64 codons, each are 3 base pairs long. There's only 20 amino acids plus 3 stop codons. Therefore, you can have some mutations that are known as synonymous, where even if you have a mutation, depending upon which one it is, it will still call for that same amino acid based on the triplet codon. Okay, so let's go over an example of a frame shift mutation. So let's say you started out with the DNA sequence towards the very top, and that codes for those particular amino acids to the right. By the way, that should make sense, and if you go back to the table, that you should be able to confirm it, all right? Let's say you have an insertion mutation where you insert one base pair, okay? That shoves everything to the right, one base pair. What you can see then is that after that first triplet codon, everything changes and the amino acids that are brought in are different. Okay, then that's one possibility. You can have an insertion mutation, by the way, that is not a frame shift, and that's if you do this, if this happens in groups of 3. So in the middle example, there it inserts 1T. In the bottom example, it inserts 3. Okay, what this does is this adds a amino acid, but it doesn't change everything from that point on. Okay, now this is a very straight up question and one for you to apply. Which one is more likely to be deleterious or harmful for the critter? Okay, so take a moment and then we'll come back together and address this. Pause the lecture right now and give it your best shot. Okay, so if we think through this, okay, and we ask which mutation is more likely to be deleterious, obviously it should be the insertion or deletion of a single or double base pair. So when you have a frame shift mutation, that's a problem because everything is messed up from that point on. However, if you have an insertion mutation which doesn't shift the reading frame and you're just adding an extra amino acid, it's not that big a deal. Oh, look, a smiley face, write this down, okay? So this should make sense that the one that's worst for the critter is the insertion frame shift mutation. If you have any questions, let me know. Okay, so we have another application question based on your reading. Okay, so what type of mutation is shown here? So what's happening is you can see the order of the genes on the chromosome. One part of that chromosome flips, okay? And then that leads to what you see at the bottom where the order of your genes has changed. Is that a duplication, deletion, inversion, reciprocal translocation, or aneuploidy? Think about it a moment and then we'll talk. Okay, so hopefully you answered C, inversion, okay? So remember, duplication means you get 2 copies of it. A deletion means you're missing DNA. And you should be writing these descriptions down, by the way. Write this down, okay? Smiley, happy face, write it down. With an inversion, you're rearranging the DNA. Okay, and so you can see that you're flipping the order of the genes. That's an inversion. If, by the way, you might be wondering what the rest of these are, then a reciprocal translocation means you're swapping DNA between non-homologous chromosomes. Okay, so let's say, chromosome arms from set number 1 bumps together with chromosome number 2 and they end up, swapping DNA. That would be a reciprocal translocation. Okay, and not last but not least, but aneuploidy means you're missing whole sets of chromosomes, while polyploidy means you're getting a complete extra set of chromosomes. All right, so hopefully these all make sense. I want you to know what these different types of mutations are, okay? And if I show you an example of a mutation, you should be able to tell me what type of mutation it is based on what we've talked about. Okay, so we have the different types of mutations we've discussed so far on the left, okay? And what I want you to do is underneath each category on the right, write which ones you feel work for this, okay? And obviously there's multiple answers and they're not necessarily mutually exclusive. So for example, you could have an indel that's also a frame shift, but it doesn't always have to be, okay? So go ahead and take a moment. This is an application question and this is really an important one. So take a few minutes to write this down and do it properly. Yes. All right, everyone. So if you've gone through, okay, and you filled this out, hopefully this makes sense. Remember, it's not necessarily mutually exclusive. You can have a frame shift mutation, which is considered a point mutation if it only ends up bumping over 1 base pair. Also, that mutation type, in order to see it, has to be in a coding region. Okay, which mutation types are also non-synonymous. All right, so this should kind of make sense. Now, I'm not going to ask you to fill this out on an exam or something along those lines, because the reality is you can argue a couple different ways for these. But at least read through these and make sure that overall they make sense. Okay, everyone, so is this a normal human karyotype? So remember, a karyotype is just a staining and placement of the different chromosomal pairs. You've got pair number 1, 2, 3, 4, and so forth, okay? Now, the thing to pay attention to is that very last set of chromosomes, all right, the XY, because what that tells you is that these are not sister chromatids. These are individual chromosomes. So you're looking at pair number 1, pair number 2, pair number 3, and so forth. Given this information, address this question, and it's not C. All right, so hopefully if you look at this, and again, you're seeing the XY, which tells you that these are homologous chromosomes, not sister chromatids. The question of, is this a normal human karyotype? Yes, yes it is, okay? And so just so you all don't panic, my goal is not to, I want you to have fun out of this class, okay? I want you to relax and enjoy it. So if I'm gonna have you address a question like this, it's gonna be obvious, like looking at the 23rd set of chromosomes, okay? I won't try to trick you. That's not my goal. I want you to learn the information, not be stressed over it. All right, so what's wrong with this karyotype of the cell? Okay, first off, there's something wrong with it. But what I want you to notice is if you look, these chromosomes are attached. Okay, so the chromosome 1, chromosome 2, those look like one single chromosome with sister chromatids. Okay, so given that information, fill out the process and the result. All right, so if we're looking through this karyotype and we know there's something wrong with it, okay, what we realize is that, you know, we have these chromosomes that are acting like chromosomes, but then we have an X and a Y. Okay, so there's a single copy of every other chromosome except the X and Y. That's a problem. The process that causes that is known as non-disjunction, okay, because you didn't have them splitting in meiosis the way they were supposed to, and this results in an abnormal number of chromosomes, otherwise known as aneuploidy. Okay, so you can have either an X or a Y in your gametes, but you can't have both, because if you do, you're going to have too much genetic information, and with humans, that's always a disaster. Okay, so I want you to practice drawing the process of non-disjunction, okay? Non-disjunction means that your chromosomes don't separate, and you get cells at the end with an abnormal number of chromosomes. So I want you to draw meiosis with two sets of chromosomes, one chromosome set 22 and the other X and Y, and I want you to show non-disjunction of X and Y. Okay, everyone, so if you drew non-disjunction, ignore the dog growls in the background. If you drew non-disjunction with chromosome 22, and I wanted you to show a non-disjunction of X and Y, which meaning X and Y don't split, okay, think about going from in the cell cycle through the synthesis phase, then you're going to go through meiosis 1 and the X and Y don't split, so they look like this, and then you go through meiosis 2, and this is what you should have. All right, if you have any questions, let me know, but ideally this should make sense and should be pretty easy. So you might be asking yourself, how often do mutations happen? And of course, we tend to focus on humans because that's the group we're most interested in. Well, the question is kind of complicated, and it depends on what you're looking at. So if you're looking at new mutations per generation, okay, then you look at the graph on the left, in which case then what critters mutate the quickest? Well, obviously RNA viruses and retroviruses, and per generation, humans don't mutate that quickly. In fact, we're one of the least ones to do so. But if you look at per genome per generation, so what this means is you're taking into account the number of genes, okay, and the number of base pairs are in there. And if you do that, then what happens is humans actually mutate pretty quickly, okay, and DNA and viruses, not so much. Because our genomes are actually much larger than that of viruses, okay? Human genomes are about 20 to 25,000 genes, somewhere around 3.3 billion nucleotide base pairs. Viruses, less than 10 genes and 5,000 base pairs, okay? Just to give you some perspective, bacteria can be between 500 genes, you know, around 500 genes or 500,000 base pairs. Drosophila, 17,000 genes and 122 million base pairs. Just kind of gives you an idea. So when it comes to the genome, that's why there's a difference, because you take into account all the different genes that are there, in which case then we mutate kind of quickly. All right, let's try another application question. Okay, and you should be able to do this based on reading of your assigned readings beforehand. So let's say you have a population treated with antibiotic. All cells isolated after the treatment were found to be resistant. Why is that the case? Okay. Is it because the antibiotics caused mutation to arise that made them resistant, or some cells had resistant mutations in them already and those could survive and reproduce? Okay, so back in the 1950s, there was a scientific team, Esther and Joshua Lederberg. They conducted an experiment. Okay, so each green circle here represents a colony, which contains thousands of bacteria that came from a single cell. All right. And they found this really cool thing where they could actually stamp plates with bacteria to get the exact same pattern from one plate to the next, okay? So they had their original plate that looked like that. They had a plate that contained antibiotics that they stamped it. This one had penicillin. Okay. Then, after they grew up in the presence of antibiotics, only colonies X and Y ended up being able to grow. Okay. And so once they took these individuals and they stamped them across all the plates, these were the only two that could grow after washing with antibiotic, because what happens is the antibiotic killed everybody else, but these guys were the only two colonies that were resistant, so they survived. All right, so what appears then, okay, from this experiment, remember, so there's the original plate, then those bacteria were stamped onto the other three plates, okay? The first one contained antibiotic, and so, that was the initial stamp, and then the third plate, they let them grow, in which case there was only two colonies that could grow in the presence of antibiotic. And then they went back to that original plate, and they washed it with antibiotic, okay? And then it killed off everybody except those two colonies, which actually this is really, really cool, okay? So what this means is that if you recall the question we had is when it comes to mutations, what happened? Well, some cells were already resistant, okay? And so it's not as if you have these mutations that, oh, I need to mutate here in order to survive. That's not the way it works. The way natural selection works is that some individuals already have good genes for different environments, and those are the ones that survive and produce more offspring. Therefore, the answer was B, okay? From a practical perspective, mutations arise randomly with respect to an organism's needs, and that should make sense. This is called the neutral theory of mutation. If you happen to be curious about this, it was first proposed by Mutu Kimura way back in the day. And, for the most part, this is what researchers have just found happens again and again and again. Okay, so given what we've talked about so far, all of this should make sense. Mutations are spontaneous and rare, but in the presence of a mutagen, you'll have more. They happen by chance during either mitosis or meiosis. They are random. They don't happen because a critter needs them. Okay, that never happens. You get variation, and this is what makes evolution possible. Now, there's a certain type of mutation that are not passed on to offspring and a certain type of mutation that is passed on to offspring. You should be able to figure that out and fill that in, okay? Then I want you to fill in the next three blanks. So phenotypic effect depends on how the mutation affects the gene. So if you have mutations in non-coding DNA, what happens? If they're synonymous mutations, what happens? If they're non-synonymous mutation, what happens? Okay, so what happens to the phenotype? Remember, phenotype is what a critter looks like. So take a couple minutes and then go ahead and fill this out, and this should make sense. All right, so somatic mutations are not passed on to offspring. Remember, we said several slides ago, those are the ones that happen in the regular body cells. Germline mutations, those are the ones that get passed on to offspring, okay? With regard to the phenotypic effects, most mutations are non-coding and therefore they're neutral. Most synonymous mutations, synonymous means it's neutral too, okay? Same amino acid. Most non-synonymous are deleterious, okay? And only very few mutations, either coding or non-coding, are advantageous. Remember, those are the rarest of all based on the theory of neutral evolution mutations, okay? Hopefully all this makes sense. Oh, look, a smiley face. You should write this down, okay? If you have any questions, as always, let me know. All right, so we are chugging right along, and it's time for us to discuss Mendel's first law, the law of segregation, which just means that your chromosomes will split, okay? So segregation means that as you form your products at the end of meiosis, your chromosomes will split, okay? And this in meiosis, you're first think about what's what and think about which is equational division and which one is reductional division. But the whole idea is that meiosis leads to equal splitting among your chromosomes, okay? And what we're going to do is use a Punnett square, and we're going to talk about probability of how you can figure out what your offspring will have based on, a given genetic scenario. All right, so let's do a prior knowledge check, okay? You have true breeding plants. Now this means that they are homozygous. Write that down. True breeding means homozygous. So homozygous for yellow seed plants were crossed with homozygous plants for green seeds. All of the F1 produced yellow seeds. So which trait is caused by the dominant allele? In other words, which one of these is dominant? Okay, that's part one. Then, if these F1 plants were allowed to self-cross, what fraction of the F2 would you expect to be both yellow and heterozygous? By the way, F1 is used to represent the first filial generation or the children, okay? F2, and this is Latin if you happen to be curious, represents the second filial generation or the grandchildren of those, parents that were bred initially. So use your prior knowledge. Hopefully you remember from high school. Isn't it great how all this stuff ties together? Address these questions, take a moment, pause lecture, and then we'll chat. All right, everyone. So as we're going through our answers, which trait is dominant? Yellow seeds. The reason you know this, of all the F1 were yellow. Okay? If they were allowed to self-cross, what fraction would you expect to be yellow and heterozygous? This is going to be C. Okay? My suggestion to you is to use a Punnett square to work out this cross and get some practice in. All right, so as they say, a picture is worth 1,000 words. Okay, so let's go through this with a picture. So you got your P1 generation. They're homozygous, which means it's big A for yellow, because we know that's dominant based on what we talked about before, and little a is green. And homozygous, so it's got to be big A, and little. Okay, now those are crossed together. You get a heterozygote, big A in your F1 generation. Okay, then what happens is you cross an F1 big A with another F1 big A. Okay. Now, the way the arrows are showing here, the deal is that if you have a big A, then in the gametes that are made from that one, you have a half a chance of getting a big A and a half a chance of getting a little a. That's the process of segregation, meaning that your gametes will, when you have a critter and they form gametes, you're going to get half the number of chromosomes, and that should make sense because we talked about it before. In meiosis, you go through reductional division and then equational division, and the reason that you do is because in your offspring, you have to have the perfect number of chromosomes, otherwise things go wrong. So you reduce the number of chromosomes by half during the reductional division stage, okay, and that's when you get the results that look here. Now, then you use a Punnett square to draw out all the different possibilities. So you have a fourth of a chance of getting a big A, fourth of a chance of getting a big A, fourth of a chance of getting big A, fourth of a chance of getting big A shown on the bottom left, and then fourth of a chance of getting little a shown on the bottom right. Okay, your expected ratio of big A, little a is 1 to 2 to 1. That's the expected genotypic ratio. You should know this. Genotypic ratio. That's the different genotypes that you're going to come up with. Your expected phenotypic ratio is 3 to 1 because anything with a big A happens to be yellow. Okay, so hopefully that makes sense. So when you get asked questions like this, you'll know how to work them through. Okay, so in this slide, we just have the notes that we mentioned before. You got your parental cross, first filial generation, what happens with the second filial generation. So there's a few other terms I want you to be familiar with, and I want you to start using this terminology now. If you ever intend on taking genetics, you've got to know this stuff. So the true breeding means homozygous. The F1 in this case are heterozygous. Yellow is dominant to green. That should make sense. By the way, this is a monohybrid cross because you're crossing one set of genes and they're heterozygous. Okay, and so the green is the recessive trait. That should make sense too. Have I mentioned you should be taking notes on your slide and writing all this stuff down because you're totally going to see this again in our class in addition to genetics. Okay, so if we're to actually take the cross experiment that we did before, okay, and we're to draw this out, what I want you to do is to be able to draw it out and take it through both meiosis 1 and meiosis 2. Okay, and as you can see here, when you do, you get a certain proportion of big A gametes and a certain proportion of little a gametes. And just looking at this diagram, you should be able to address this. The other thing I want you to think about is to remember which division is the reductional division and which one is equational division, okay? And then be able to follow the S or the synthesis phase, realizing that that's when your chromosomes duplicate and start gearing up for meiosis. Okay, so if we go through, okay, and we write down what's going on, we know that in meiosis 1, our homologous chromosomes separate. In meiosis 2, our sister chromatids separate, so the proportion of gametes that are big A is a half, and the proportion of gametes that are little a is a half. And once again, this is the law of segregation because they're splitting. So now that we know this, okay, Mendel's law of segregation, we can kind of get some predicted outcomes. So for example, if you have two heterozygotes, big Y, little y, cross with a big Y, little y, you can use the Punnett square. Realize too that the dominant is always capital, and you need to do that always, okay, at least for the purposes of this class. And then once you take genetics or move on, they'll start with this and then probably expand on it a bit. And then the symbol for the recessive is always the lowercase y, all right? Okay, so if we were to label these further, we know that the germ cells are there. We know that the sperm and the egg is there as labeled. We know the gametes are there and the offspring. And again, I apologize for the spacing. And oh, look, a green smiley face. Then we know our predicted genotypes and we know our predicted phenotypes. And again, this is hopefully all review and should all make sense. And I just want you guys to be able to think through all of this and make sure that everything makes sense. Okay, so rather than working a whole bunch of Punnett squares, there's actually other ways to address questions like this, okay? And it's based on the laws of probability. And in case you're curious, the laws of probability are why you should always stay out of casinos. Okay, because when you figure out the probability and the likelihood of actually, having something happen, well, in casinos, you realize that the odds are always stacked towards the house and against you. Okay, but when it comes to probability and genetics, you can kind of figure out what offspring you're going to have without even having to use a Punnett square. Now, if you're curious, the Punnett square was initially invented by a man named Reginald Punnett, who wanted to use a simple Punnett square to just illustrate Mendel's laws. Okay, but they were never meant to cross anything more than a single set of genes. And I don't ever want to see a huge giant Punnett square on anybody's papers because that just gives me a headache just thinking about it. So instead, you can use what's called probability to figure out what the genotypes are predicted and what phenotypes you would predict given a particular cross. Okay, so there's a couple different rules you need to know while working through probability. Okay, so if you see the word and, you multiply. So both are and, you multiply your numbers together. If you see the words either or, you add. Write this down somewhere, okay? Now, let's actually work through this. So now what I want you to do is move to the bottom left where you see P of big Y, big Y. So let's say you're given the cross, okay, shown above where you have two heterozygotes and you want to know the probability of having big Y, big Y. All right, so then what you do, so basically you want to know the probability of having a big Y from one gamete and the probability of having a second big Y from the other individual. Well, we know based on the law of segregation that you have a half a chance of getting a big Y from one individual and a half a chance of getting a big Y from the other individual. And because it's a half a chance and a half a chance, you multiply, which gives you 1/4. Now take that number and go double check it on the Punnett square. Do you have 1/4 for big Y, big Y? In fact, we do. Next, you want to know the probability of big Y, little y. Okay, so once again, you work out the probability of the big Y, you work out the probability of the little y. That's one possibility, okay, and it's and right there. However, then we go with or, because then you can have big Y, little y from the other one. When you use or, you add, okay? So it's a half times a half plus a half times a half, and that gives you 2/4, otherwise known as a half. Go look at your Punnett square. 2 out of 4 big Y, little y? Yes, in fact, they are. Okay. Now, the last one, the probability of little y, little y. Okay, so it's little y and a little y. So whenever you see and, you multiply. Okay. So it's a half times a half, which gives you a 1/4. And then go double check on your Punnett square, and lo and behold, probability of little y is a 1/4. Okay. Now, the probability of being yellow. So either big Y or big Y, little y. When you use or, you add, okay? It's a 1/4 plus 2/4, which gives you 3/4. That's your phenotypic ratio of 1. Then your second one is probability of being green, which is 1/4, so it's 3/4 to 1/4, okay? So what I'm wanting to do for this to illustrate is just let you see that there's other ways to solve the same problems. I honestly think probability is easier. In theory, you could use probability to address a cross with like 5 genes, okay? Do not use a Punnett square for that. Use the laws of probability. It is much, much easier and much more straightforward, and then you won't pull out as much hair. Okay, so working the next problem. So I want you to read through this. Go slow, okay? What's the first thing we do when we see a word problem? We don't panic. But oh, look, for those of you wanting to go to medical school, this is an MCAT question. Okay, we have faculty here that write questions for the MCAT, and so this would be something useful to know if in fact you want to be a doctor. Take your time, pause lecture, work through this, and then we'll talk. Okay, so now let's read through this and make sure you guys got the same answer as I did. You have this recessive skin condition that can lead to cancer. A woman homozygous for the dominant allele. Don't let that trick you. That just means the non-cancerous version. And you know that that's dominant because we said the cancer version up above is recessive. Therefore, the woman is big A, big A. That should make sense. Homozygous for the dominant, okay? And this is all a part of learning the, truly learning the material and not just spitting it back out at, the instructor. Homozygous for the dominant allele means she's big A, big A. And a man who is homozygous for the condition, which we said the condition is recessive, therefore he's little a, okay? They wish to have a child. What's the probability the child will get this particular disease? All right, so if you have a woman that's big A, and she has children with a man that's little a, remember, this is a recessive disease. So all of the children are going to be heterozygous, meaning they're big A, which means nobody has the disease. Okay, so the answer to that is 0%. All right, so hopefully that makes sense. If not, let me know. Make sure you're comfortable with this. All right, now we're going to chug right along and go to Mendel's second law, otherwise known as independent assortment. Lots of fun stuff with us. All right, just so you all know, I love genetics problems, okay? When I was in grad school, I actually GSI'd or TA'd for genetics at Illinois, and so it's one of my favorite classes. So you guys are gonna see genetics problems, and you're gonna do so many of them, you're probably gonna be doing them in your sleep because I want you comfortable with this. Practice makes perfect. So we're gonna run an experiment now with 2 traits. We're gonna look at the seeds. So you plant grown from a true breeding, what does that tell you? That's right, homozygous for yellow and wrinkled seeds. Okay, then you're going to cross that with a plant from true breeding, once again homozygous, green and round. Okay, now you might not know at this particular stage what's dominant, but you will once you hit that F1. So you look at the F1 and you see, all right, what traits are expressed? Yellow and round. Therefore, yellow and round are dominant. Now you can go back and designate your letters for the 2 parents, okay? And hopefully that will make sense. All right, then what happens is you're going to conduct a dihybrid cross with the F1 or first filial generation. So dihybrid, di means 2, so 2 genes, and hybrid meaning you have 2 heterozygotes, okay? So you're going to cross an F1 with an F1. And when you do this, the expected phenotypic ratio is 9:3 to 3:1. All right, so this means it's complete dominance, by the way. There's no epistatic interaction, so your genes are not interacting, and you might be asking, how in the world do they know that? Well, on the next slide, we're going to break everything down and show you in a Punnett square. What I want you to be able to do, though, is think through, if you were asked to do this, okay, using probability, could you solve for the same questions? All right, so once again, let's take this cross and actually, with both traits, and actually work through a Punnett square, okay? So again, it's a dihybrid cross. You've got 2 traits, all right? And the deal is that for each of the parent, okay, you have 4 different potential gametes. Big A, big B, big A, little b, little a, big B, little a, little b. Okay, and so those are the pollen genotypes across the top, and the ovule genotypes are the same going across the side. Whenever you work across like this, make sure you keep the similar ones close to one another. It just makes life easier. Then what you do is you use your Punnett square and you combine your pollen and your ovule genotypes, okay? So your first one is big A, big A, big B, big B. Second one, big A, big A, big B, little b, and just follow through, okay? Whip out a blank piece of paper. Make sure you can do this, okay? Realizing that there's 9 possible genotypes and 4 possible phenotypes, and the phenotypic ratio is going to be 9:3 to 3:1. I want you to be familiar, by the way, with that those fractions, 9:3 to 3:1, because we're going to come back later in the term and talk about when you do crosses like this, but your results don't look like this. And that means that your genes are interacting or things are more complicated than they may seem. And so for right now, just lock it into your brain. If you have a dihybrid cross with complete dominance, you're running this cross, you'll have their phenotypic ratio should be 9:3 to 3:1, and if it's not, there's something else going on genetically that's interesting. Now, what this means, by the way, is that if you were, and how this relates to independent assortment, is that how one set of chromosomes lines up on the equator during meiosis is completely independent of how the other set of chromosomes lines up on the equator. So, for example, you have your cross, big A, big B, little b, goes through the S phase, looks like this. Then that's going to go through meiosis, okay? 50% of your cells will look like what's above, 50% of your cells will look like what's below, okay? And the reason that happens is how set chromosome for A lines up on the equator is completely independent than set B. That's what I want you to do, okay? Stop the lecture, go through and carry this out. Draw meiosis 1 and then draw meiosis 2. It's worth your time to do this, by the way, so please take the time to go ahead and do it. All right, everyone, so if things went well, which I'm hoping they did, your drawing should look something like this, okay? If you have any questions, let me know, but this should make sense if you're going through and drawing everything properly the way it's supposed to go.